Are you scared? I'm just kidding. Do you want me to introduce you?
Okay, we're gonna go ahead and get started. All right.
Thank you, Miss Stephanie.
Hi, my name is Haley Woodward. I am a rising senior at University of North Carolina, Wilmington, and this summer I had the pleasure of working with Dr. Samar Hamad as well as Dr. Titus Reeves. My project was the role of sphingosine 1-phosphate and polymorphonuclear leukocyte, or PMN, functions. PMN are immune cells that are the first responders to tissue injury. These cells have the ability to infiltrate into tissues, release cytokines along with other toxic substances to eliminate foreign bodies and that cause disease. When PMN infiltration becomes persistent and dysregulated, this can cause substantial damage to good non-disease tissue, as is the case in inflammatory bowel disease. Inflammatory bowel disease, or IBD, primarily consists of ulcerative colitis and Crohn's disease. Both of these are characterized by massive PMN migration into the intestinal structure, but ulcerative colitis is localized in the large bowel as well as the rectum, whereas Crohn's disease is can occur anywhere from mouth to anus, but is localized in the terminal ileum. Crohn's disease also has the presence of granulomas, which are epithelial nodules containing other immune cells. Here's a cartoon of the process of neutrophil transepithelial migration. Here you can see the PMN in the bloodstream, and they will traverse the epithelium into the lamina propria, migrate through the lamina propria, and punch holes in the basement membrane here. Next, they will squeeze through the tight junctions of the epithelial cells to arrive on the luminal side of the intestine. This is a multi-step process that involves a lot of cell signaling and everything, but for our purposes of our experiment, we focus simply on a neutrophilic response. Sphingolipids are structural components of cell membranes and have emerged as key signaling molecules. In particular, they've been shown to affect cell growth, differentiation, proliferation, as well as apoptosis. And this class of signaling molecules, namely sphingosine kinase, which leads to the production of sphingosine 1-phosphate, has been shown to interact with immune cells to induce inflammatory cascades at the sites of tissue damage. Tumor necrosis factor alpha is a cytokine that has the ability to increase expression of S1P on epithelial cells. This increased expression results in adhesion of PMN to epithelial cells, thus affecting PMN infiltration into tissues. The activation of SK plays a role in the signaling responses to TNF-alpha, and the ability to promote adhesion has been shown to be mimicked by S1P. There have been several studies detailing the role of S1P on monocytes. For example, monocytes have the ability to release S1P upon activation, and this promotes inflammatory cascades at sites of tissue damage. Studies also show that S1P can facilitate PMN infiltration into tissues. However, the molecular events that modulate this have not been clearly defined. Thus, we investigate the role of S1P in PMN adhesion as well as transmigration. To do so, we performed 3 experiments after the isolation of PMN. We did immunofluorescence, cellular adhesion, as well as transmigration assays. For PMN isolation, we separated these cells from the whole blood of healthy volunteers and resuspended them at a concentration of 5 × 10⁷ cells per milliliter. And the number of PMN in the sample were counted by NPO assay. For our immunofluorescence, the isolated PMN were fixed as well as permeabilized and blocked and then labeled with a SK and FITC antibody, and then the nucleus was stained with rubidium iodide. For our cellular adhesion assay, a 96 well plate was coated with fibrinogen, which is a CD11B ligand that promotes the adhesion of PMN. And after blocking and washing, our cells were added once they were combined with our various reagents, which were an SK inhibitor, or DMS, LY kinases, which is a QI3 kinase inhibitor, MAP kinase inhibitors, as well as genistein, which is a general tyrosine kinase inhibitor. Finally, our transmigration assay, we isolated PMN and added them to the upper chamber of a transwell apparatus, and then allowed the PMN to migrate toward FMLP, which is a bacterial-formulated peptide, for 90 minutes. The results of our immunofluorescence showed that SK is expressed by PMN. Here in the no primary, you can just see the nucleus. And in our control, you can see here in the red is the nucleus, and the green around is the SK expression. You can also see this expression in the FMLP treated cells, but we did not notice a huge difference between the expression of SK in the control and FMLP treated cells. The results of our adhesion showed that here in the control of the applied load, we had about 25% of the cells adhere to the fibrinogen, and about the same was seen in our SK at 1 and 2 molar solutions. And up in the corner here is what the 96 well plate looks like. And at the bottom, we saw that genistein didn't have much adhesion. It was only about 8%. Here is a cartoon of what the transfilter apparatus looks like and how they migrate towards the FMLP. Here in the top well you have the PMN, and they're in Hanks Plus. And then over the 90-minute period, they're able to migrate downward towards the reservoir, and that has the FMLP chemoattractant. The results of our transmigration showed, well, here in the top is a control of what normal PMN migration looks like. In the first 30 minutes, you have an increase in PMN migration towards the chemoattractant, and that's steady through the second half hour and tapers off towards the third and final time interval. In genistein, you see a huge increase in PMN migration within the first 30 minutes, but it's not steady through the second and third 30-minute time intervals. And with our SK inhibitor, both 1 and 2 molar solutions, we saw that there was a delayed migration, which occurred mainly in the second 30-minute time interval. So in summary, S1P is expressed by PMN, and FMLP treatment did not result in increased expression. SP is determined by treatment with SK inhibitor, has minimal effects on PMN adhesion to fibrinogen, and inhibition of SK has delayed effects on PMN transfilter migration. I would like to acknowledge my mentor for the CERT program, Dr. Samar Hamad, as well as my mentor, Dr. Titus Reeves. He graciously let me in his lab, and his lab technician, Anita Smalls, who taught me everything I know, and I can't thank you enough. And also the medical students in the lab, Jacqueline Savage and Megan Anderson, along with the